Hi, everyone, and welcome to today's live webinar on AI plus healthcare, revolutionizing drug discovery and development, part of the AI plus healthcare series by AI Certs. My name is Rohini, and I'm delighted to be your host for the exciting session. If you've ever wondered what is AI Certs, let me give you a quick introduction. AI Certs offers role-based AI and blockchain certifications aimed to fostering lifelong learning. Our mission is to help organizations build smarter, future-ready workforces, adapt to change, and drive growth. And here's a big vision. We are on a mission to certify 1 billion people worldwide. Let's take a moment to watch a brief video and discover more about AI certs. I request my team to please play the quick YouTube link. And yeah, it's just a small, quick video. We will also be posting this on our chat boxes.
The only constant is change. Staying ahead means constantly adapting, evolving, and growing. You need the right skills to thrive. Artificial intelligence and blockchain are revolutionizing everything we do, from how we work, create, and solve problems, to how we secure data and build trust in a digital world. Do you have the skills to thrive in this new era of AI? To stay ahead, you need the right tools and knowledge. But with so much information available, where do you start? AI Certs empowers you to equip yourself for the future with industry-recognized, role-based certifications in AI and blockchain. Whether you're an individual looking to upskill and advance your career, or a business seeking to empower your workforce with cutting-edge AI and blockchain expertise, AI Certs is your trusted partner. Our certifications are crafted by a team of leading industry experts, ensuring they align with the latest trends and real-world demands. We understand that everyone learns differently. Our flexible learning options allow you to study whenever and wherever it works best for you. With a tech background or without one, our comprehensive range of role-based certifications equip you with the in-demand skills you need to succeed. Stand out in the job market with recognized AI and blockchain credentials. Leverage the new technology to become more efficient, innovative, and impactful. Help your company thrive in the AI era. AI Certs, empowering individuals and businesses to lead the AI revolution.
Well, okay. The intersection of AI and healthcare is transforming drug discovery. Did you know that? Like making it faster, more efficient, and far, reaching in its impact. To explore this exciting topic, we have with us Venkatesh Mysore, a pioneer at the forefront of AI driven healthcare innovation. Before we dive into the keynote, here are a few housekeeping details. You may share your questions anytime using the Q&A box. We'll address them during the Q&A session at the end. The webinar is being recorded and we will share the recording with you after the event. Now, let me introduce our esteemed speaker. Venkatesh Mysore is the CTO and co-founder of ICOM, INC, a deep learning driven protein engineering company headquartered in Berkeley, California. At ICOM, Venkatesh is his and his team are leveraging patented generative language models to design trillion protein libraries synthesized and screened using their proprietary Yota display platform. ICOM's cutting edge pipeline focuses on GPCRs and their collaborations span ion channels and transcriptions factors, addressing critical challenges in the therapeutical areas such as oncology, immunology, and metabolic disorders. Venkatesh's work at the intersection of AI and protein engineering embodies the future of healthcare innovation, and we are thrilled to have him here to share his insights. Ladies and gentlemen, please join me in welcoming Venkatesh Mysore.
Hello to everyone and the AI Certs community who've dialed in from various parts of the world. My background and why I grabbed this opportunity to talk here is because my career, I made some unusual choices with my foundations in computer science. I've opted for a career that pulled me closer and closer to molecular and cellular biology. And then that's where I discovered that there is a lot of work that can be done by using the power of computation to actually discover and develop new drugs. So I've been in the space of computational drug discovery for over 15 years between DE Shaw Research, Atomwise, and NVIDIA. And now at IKEM, we are engineering our own class of protein biologics using a novel deep learning based approach tied with ultra high throughput synthetic biology. But I'm not here to pitch about IKEM, but instead use this opportunity to tell you how the field, as I've seen it over the past 2 decades, how it has evolved and where it is headed, the field of computational drug discovery. So to set the scene, let me share a few slides. I don't want to dwell on the slides too much, but it'll help get all of us on the same page. So to talk about AI's journey in drug discovery or in a drugs journey, let's start with the part that actually touches the patients. So we as patients, a certain medication comes to us, but how is AI used there in that very, very last stage after a drug's approval? So it turns out that if you are a patient and there is a newly approved drug, you don't automatically benefit from the drug. A doctor has to be willing to prescribe that medication. So there is a lot of computational study that goes on to identifying the patients who are likely to benefit from the newly approved drug, and then the doctors who are likely to prescribe those medications to those patients. So here, there are many companies that have brought new uh, technologies such as large language models and other breakthroughs in deep learning to reason about patient electronic health records to to sort of impute the values: who is likely to be a patient, who is likely to benefit from the treatment, which doctor is likely to prescribe this medication, and how do we target that patient population or that doctor population by advertisement, conferences, and such. So this is also the use of AI in ultimately delivering the drug to a patient. But this is not going to be the focus of today's discussion. So let's move one step uh, further back in the journey of a drug. So before a drug can get approved, the biggest question is the clinical trials. As you're all probably well aware, clinical trials happen in three phases where they establish safety, efficacy, and then ultimately efficacy on a much larger scale. So this is what I was starting with, saying that after a drug is approved, there is a lot of AI that goes into. Today, there are many companies applying sophisticated reasoning to ensure that the drug reaches the patient. Because only if it is financially viable can the next drug discovery project happen. So, in the previous step, to get that drug approval in a clinical trial, once again, a lot of analysis is required in identifying the correct patient cohort and being able to recruit those patients for the study. So once again, we go back to electronic health records, and then a little bit more into a person's biological profile. As you're aware, starting the year 2000 or so, the Human Genome Project made it possible to now very rapidly establish a connection between certain genes that are part of our genome and certain clinical outcomes, such as response to medication, suitability for a certain treatment. For example, let's say you're targeting a certain mutation of a certain protein in cancer. You want to do a genetic screen to ensure that that person, in fact, has that mutation. It could be part of their genome, or it could be just part of the mutated genome at the site of the cancer. So here, once again, sophisticated analytical tools are employed, all under the AI umbrella, to predict, to recruit, and to conduct these clinical trials. And there is a whole marketplace that has opened up where many more hospitals, how to bring many more patients into the pool. You would think that by 2025, these are solved problems where every hospital anywhere in the world can participate in a clinical trial, but it is not so. There are so many technical challenges, there are so many regulatory challenges, and of course, things like patient privacy: who wants to participate in a trial, who owns the medical records. So these two buckets of what happens during clinical trials and what happens after a drug is approved fall outside the purview of today's discussion, but they are nevertheless fields that are very rapidly giving a return on investment, if you will, from a business point of view. But now let's go one step further back in a drug's journey. What happens before phase one clinical trials? So, phase one clinical trial is when the drug is dosed to a human being for the first time, first in human. But realize that clinical trials are yet another experiment. Every stage is actually an experiment. And before we dose a human with a drug, we want to be absolutely sure that the drug is likely to work, but even more importantly, that it is safe to be administered to a human being. We are all aware of toxins, we are all aware of venoms, we are all aware of off-target effects, toxicity. We've heard of occasional adverse events, extreme adverse events in clinical trials. So, how do we predict what is going to happen in a human body? So it turns out that even with the world's best supercomputers and all of NVIDIA's GPUs, you still cannot model and simulate a human being's body. So large mammal with organs, tissues, interconnections is just simply beyond today's AI or computational revolution. So experiments cannot be bypassed yet. We are not yet in the phase in human history where we can say we can bypass experiments and develop drugs entirely computationally. It sounds nice as a claim, but it's unrealistic as of today. So, what is the best possible predictor of what's going to happen in a large mammal such as a human being? Well, the best possible predictor is what's going to happen. What happens in a small mammal such as a rodent or a pig or a monkey of some specific criteria. So, what here, how is AI or how is computation more broadly used? So, this is a bucket where we actually are severely lacking. For clinical trials and patient medical records, I would say there is a very well posed problem, and there is a large corpus of data. Whereas for the problem of predicting what happens to drugs in mice or in a small mammal, we are still collecting the data. So some startups have mushroomed where they have tried to multiplex data collection within animals. We now have ultra high throughput microscopy, other ways of measuring what happens. But this is still very early stage. We are still modeling what happens in an animal's body. But there are certain well-defined sub problems that we are beginning to predict, such as toxicity or immunogenicity, where we know one or two off targets that it shouldn't bind to, and so we can very quickly check if it binds to that. Similarly, we want a drug in some cases to remain in the animal's body for longer so that it has a greater therapeutic effect, such as protein based drugs. Or there are others where we want it to do its job and then get flushed out of the system so that it doesn't accumulate in the wrong organs in the body. So, some of those properties can be predicted, and deep learning and other technologies are beginning to be applied successfully there. But now comes the part that I want to spend a good amount of time today sort of telling you about how this field has evolved, and that is the actual job of discovering the drug. So you want to come up with a novel chemical entity, a molecule that did not exist before, that solves a problem. So this is the hardest part in some ways, because how do you find something that does not exist? Maybe improving something that previously works, maybe you have a starting point, but how do you actually uncover something that does not exist? So this is the field of drug discovery and development, and there are plenty of breakthroughs here in AI that I'll shortly be sharing that have changed how we do this in the last 2 decades. But I accidentally misspoke. I described this as the hardest problem in the pipeline, but the even harder problem is actually understanding what goes on in the biology. What is the root cause of the disease? What is the target protein that I need to block in order to treat a disease? So, uncovering that disease biology is actually and forever will remain the biggest challenge, because our body and the biology, both at the molecular level and at the physiological level, is so complex that we cannot very trivially model using any amount of bioinformatics or systems biology. They take us so far, but really beyond a point, We need to do actual experiments. We need to collect lots of data about disease. And there are many companies who dwell on target discovery, finding the protein that one ought to go after rather than focusing on uh, the discovery of the drug. Because for that, there are at least some approaches. Whereas for understanding and uncovering new biology, the kind that wins you a Nobel Prize, but also gives you a new opportunity to treat a disease. That's what the future is all about. So to now, let me get deeper into this particular bucket of drug discovery and development, focusing on the earlier stage because that's where AI has already made a very dramatic impact. So I mentioned that the first step, which is the hardest part, is knowing which target to make a drug for. So drug discovery is ultimately about going down to a cell, and cells have proteins, and you're usually trying to stop a protein from doing its job. So why would you want to stop a protein from doing its job? Well, that could be because that particular protein is overactive, or maybe it has a mutation that makes it do something it is not supposed to, so you're trying to suppress it. So this is where the biologists have to uncover which protein is responsible for the disease. So uh, rather than use slides at this point, I want to make this connection that to uncover new biology, the answer is actually not in computation. The data has to come from somewhere. And so companies, for example, you have in vitro, you have companies like Recursion, where they invest a lot in ultra high throughput experimentation. So trying to get genotypic data, phenotypic data, trying to tie what happens at the molecular level with what happens at the large scale level. So one good example is, let's say you had cells, and you in a cell you were able to grow, let's say, a cancer cell line, and you are trying to find out which compound could potentially treat that particular cancer. While you cannot do an experiment on a patient, you can do these experiments on the cancer cell line. So you can grow these cell lines, and then you can administer several different drugs, let's say, that you have in your in-house bank, and then you can see which cells begin to look healthy. So this is how we were doing it some time ago. But now with robotics, what I just described can be entirely automated. So what you can do is you can have 100,000 to a million different petri dishes in which these cell lines are being grown, and inside each cell line, inside for each petri dish, you can imagine administering a different compound through robotics, and then you can imagine employing ultra high resolution microscopy to take images of these cells. Then that's where AI comes in. So up until this point, it is all about scaling, robotics, and miniaturization that has been possible through breakthroughs in or advances in molecular and cellular biology. But what do you now do with those images? So that's where deep learning over images comes in. So as you're all well aware, deep learning over images was actually developed for other applications such as graphics, such as reasoning about the real world in the context of, let's say, self-driving cars. But quietly but surely, those same technologies have been applied very, very successfully for reasoning about medical images such as X-rays, MRIs, and now it is all about digital pathology. So you can now imagine training an unsupervised representation learning over high resolution images generated from microscopy of cells. So from that, you have uh, a way of representing biology as numbers. So the moment you can convert your input image or input audio or input text into numbers, that's when reasoning can begin. So you need a representation of cells or images of cells. And so by applying very similar techniques to what has been applied in self-driving, we now have technologies for converting this representation of cells, over which you can now do the reasoning of, does the cell look like a healthy cell or does the cell look like a sick cell? So this is the transition that we want to induce. We want to uncover compounds that seem to make a cell that resembles a sick cell resemble a healthy cell. So this is phenotypic screening on an ultra large scale, aided by robotics, microscopy, and of course deep learning over images. So now let me go on to the next step, and talk about the uh, other application, uh, the next step after target discovery. So that's where we have, uh, what happens to a target? What does a target look like? Where on the target should I bind? So now we are moving from target discovery. So you've uncovered the protein that you're supposed to block, but now you need a strategy for how to block it. So you need to know where on the target protein you need to act. So a protein is a large three-dimensional molecule. It has many different phases, many different interfaces that it uses for interacting with other cellular partners, which could be proteins, which could be RNA, DNA, which could be other cellular small molecules, or it could have a catalytic site. Proteins that catalyze the reaction are called enzymes. So it could have a catalytic site or an active site or a pore where it conducts ions. So so you need to understand where on the protein you need to somehow interfere and block to achieve the blockage of function or the reduction in activity that you're ultimately trying to achieve. But now to understand where on the protein you need to make a change, you need to do a completely different set of experiments and a completely different set of reasoning. So this is where, to understand where on the protein you need to operate, previously what we were doing was we were hoping to get the three-dimensional structure of the protein using some established technologies such as X-ray crystallography, NMR spectroscopy, or more recently cryo-electron microscopy. But they all have limitations. Only under certain conditions and for a very tiny fraction of the proteins in the real world can you actually obtain the three-dimensional shape and structure. And to make matters worse, roughly 50% of the proteome is inherently disordered, meaning it is structureless. So as a result, there is only so far that experiments can get you. So then came the big revolution in protein structure prediction, which you may have heard of as AlphaFold. This started in a company called DeepMind, subsequently acquired by Google. And we all know that last year, this particular breakthrough actually won the Nobel Prize for protein structure prediction. So now the question is, how does protein structure prediction work and how does it help? So first off, let us understand why it helps, which is you're trying to make a molecule, the drug, that has to go and bind to a very specific point on the target protein. So you need to know what the target protein, the binding site looks like. And for that, you need to know the three-dimensional shape of the target protein. So that's the utility. But now how are we able to predict it? So before AlphaFold, we would do, I would say, a more commonsensical approach, which is look at similar proteins. And from their structure, let's say we had solved the structure of a similar protein using X-ray crystallography, we would say, hey, that protein looks like this. This protein is different in the following ways. So maybe its structure will be different in the following ways. So that was called homology modeling. And that's where that's what we were doing till about 10 years ago. Then slowly, deep learning started to be applied. So we are now reasoning about protein structure, but using some clever tricks. So I'll summarize what some of those clever tricks are. So previously, to guess the structure of a protein, we were only able to use the structures of proteins that were similar. But the reality is there are millions and millions of proteins in nature whose structure we don't know, but we know their exact chemical composition, also known as their primary sequence, which is the linear description of the chemicals that form a protein. So protein can be thought of as a chain of chemical beads, and each chemical bead has 20 different flavors, naturally occurring amino acids. So instead of just using the structures of similar proteins, AlphaFold and subsequent methods started incorporating the sequence level data about all similar proteins. So we may not know the structure of a similar protein, but we know its chemical composition. And the reality is the chemical composition of a protein is actually very largely constrained by what it ultimately needs to do, which is its function, and the function is very often enabled by its three-dimensional structure. So this knowledge about protein structure is actually encoded in its protein sequence, and therefore we started reasoning We started doing better when we were able to reason not just based on the structure of similar proteins, but also on the sequences of similar proteins. So that is one of the breakthroughs. Second breakthrough is very, very closely tied to how to methods that were developed for reasoning about the three-dimensional world. So objects have an X, Y, Z coordinate. So how do you represent three-dimensional structures on a computer? Then the question is, a three-dimensional structure, if you rotate it to the left or to the right, the structure actually doesn't change, but its XYZ coordinates will be different. This can confuse a model. So one of the breakthroughs, or one of the key ideas incorporated in AlphaFold, is in the model, the way you represent information, it should be it should not represent a molecule differently just because it is rotated in XYZ. So because a true protein or any three-dimensional object, just because you rotate it or move it around, it doesn't become a different object. Its properties are in fact uh, remain the same. So these two, I would say, are two breakthroughs, or two novel ideas that were incorporated into the AlphaFold prediction algorithm, which is subsequently improved into AlphaFold 2 and AlphaFold 3, which can now predict protein structure prediction along with how it interacts with other proteins, with small molecules, with RNA, DNA, and nucleotides as well. So this is the big picture, that to achieve what we are trying to do, which is drug discovery, we need to first know what protein to target, and then we need to know where on the protein we need to bind a molecule. So this is where we need protein structure prediction. But what happens after that? That's where we need to know what chemical uh, matter will actually stick to the protein on the site that we have identified. So we have found the target protein, we've found the binding site. Now we are trying to find which chemical molecule should I use to go and block that binding site. So imagine a cavity on a three-dimensional surface, and you're trying to fill in that cavity. In fact, most drugs that we take, the over-the-counter pills that we pop, let's say for a simple headache, ultimately many of them work by binding to a very specific three-dimensional cavity on the protein surface, thereby most often disabling the target protein from achieving its function. So now the question becomes, what are the different darts that I can throw that will go and strike the target? Meaning, what are the different chemicals that I can make? So here there are broadly 2 or 3 different classes of biomolecules that we should be aware of. So most often, the most common kind are what are called small molecules. So when we say small molecules, they are small with respect to the size of the protein. These are anything from your Tylenol to a common medication that you take for cold or cough or fever, anything that you often pop in a pill, that's usually a small molecule. So these molecules work, like I said, by blocking the binding site on a target protein. And how has AI changed the way we do this? So previously, when we were doing small molecule drug discovery, the idea was there would be a small, finite uh, universe of chemicals that one could buy. This would be a compound catalog. And from within that compound catalog of, let's say, a few million compounds, we would take each of those compounds, try to computationally place it where the binding site was, and then come up with a physics inspired score. Because remember, ultimately, the interaction between a drug molecule and a target protein is governed by the simple laws of physics. So if you can actually plug in those forces between the atoms, everything else follows from that. So to take a slight tangent here, I want to talk about a field called molecular dynamics simulation, which was founded on this exact principle, which is, if you can capture the atomic forces between atoms, then really all of biology should naturally follow from that. So this has led to companies like DE Shaw Research investing in supercomputers that turn a computer essentially into a microscope through which proteins and protein drug interactions can be observed. So this has led this has fundamentally improved our understanding of proteins, and we are now able to computationally see and explain some of the measurements that we take in fields like nuclear magnetic resonance spectroscopy. So this is one way to go about it, but it's very expensive and it cannot reason about everything that matters. And ultimately, it is also very slow. You have a throughput of thousands or hundreds of compounds depending on your computational budget. But we now have compound catalogs not in the millions, but actually in the billions. So now two new problems have been solved with the use of AI in small molecule drug discovery. The first problem is the compound catalogs themselves are no longer millions long, but billions long. So even if you develop a sophisticated, your old scoring function that you had, which was based on physics-based principles, was slow and approximate and could not do billions of compounds. So there are two or three different incremental improvements that have happened in the last decade. First off, rather than using an entirely physics based or a statistics based scoring function, we have now developed multiple deep learning based scoring functions. So, what do you mean by a scoring function, and how does deep learning come in there? A scoring function is a function where given a target protein and the biological molecule that you're making, let's say a small molecule, given those two as input, it needs to predict a quantity called ligand binding free energy, which can simply be thought of as how strongly will this drug molecule bind to the target protein. You're just trying to predict binding. So now for this, rather than use physics-based approaches and statistics of what compounds worked in the past, what the new thinking is to first do representation learning on protein. So you understand the inherent chemical language of proteins. Similarly, you do representation learning on small molecules. So you understand, you have a representation of small molecules that already knows the rules of chemistry. It has learned that in unsupervised manner because it has seen only uh, feasible chemicals that have been synthesized before. So by knowing, having a representation of the chemical space and having a representation of the protein space, one can now develop models where you can go to literature from various different databases and pull out how proteins and small molecules interact. And from that, you can convert, you can frame a supervised learning problem where given proteins and small molecules, each with its own representation and a label, whether they bind or not. Sometimes you even have the annotation of how strongly they bind. With this triplet, protein, small molecule, and some annotation about the strength of binding, you can now build a deep learning model or even a classical feed forward neural network. It doesn't have to be very deep because the deep learning has already been used to learn the representations of the protein and the small molecule. You can now build a classifier or a regressor that can predict the strength of interaction between these two classes of biomolecules based on annotations that you have developed inside your company or that you have pulled from public databases. So, this is how deep learning has impacted or initially impacted small molecule drug discovery. But now, in the last 5 years, we have moved on from methods that are sophisticated scoring functions inspired by deep learning, maybe clever ways to reason about billion compound spaces. So you do some intelligent traversal of chemical space. So that is one way of solving the problem. The second way of solving the problem is to say, hey, give me the binding site and I will, like an expert chemist, actually construct a molecule that will fit in rather than looking at a long list of guesses and seeing which one fits in. Maybe I know enough to be able to directly design a molecule that will fit perfectly into that pocket, into that binding site. So, this approach is called generative small molecule design. So, as an input, what generative models do is they get the binding site represented in some clever way using representations from geometric deep learning, which is a way of encoding three-dimensional spaces. One can then have a model that essentially spits out one after the other molecules that it thinks should fit in perfectly into that pocket. Well, it doesn't work this simply. Drug discovery has not been solved, but we now have plenty of models, and there are being improvements reported not every year, but almost every month. There is a breakthrough paper, or each paper claims to be a breakthrough or a substantial improvement over the prior month's work, where we are now able to generate molecules that really score very well on a computer when they are tested against the protein. But how do they fare in the real world? So that's where a different problem comes in. Most chemicals that you draw on a computer or on a piece of paper are actually very, very difficult to synthesize. So synthesizing a small molecule, if it's not in a some standard format where the composites already exist, can cost upwards of $10,000. With some projects failing to failing to synthesize a molecule even after $100,000. So synthesis of novel small molecules is actually an extremely hard and unsolved problem. People are making improvements in predicting synthetic feasibility and using that to generate molecules that are, ahead of time biased to be synthetically accessible. This is all ongoing, but generative small molecule design has now taken the world by storm. But it's not as though it has reached the clinic yet. There are so many stumbling blocks, but we can say that it is a very exciting space, and we are all, as computational chemists, waiting to see how these translate to the real world. But so far, I've been talking about small molecule drug discovery. The other aspect is protein design. I'll save that for now since I have one slide to talk in greater depth about protein engineering. But before we go to protein engineering, let's quickly complete the journey of a drug and what it needs to do, what a drug developer or a drug designer has to do on a daily basis. So you've picked the target, you've found the binding site on the target, then you've used, let's say, in the context of small molecules, you found one particular small molecule. Maybe you did generative small molecule design, or you did intelligent navigation of chemical space from a billion compound catalog. You found some molecules, then you sent them to a contract research organization, or you sent them to your own chemistry department. They did the assays, something works. But does the development of that molecule stop there? No. So what you've really done is only uncovered the first molecule, which is often called the hit. So you now have something that fits in there. But now it needs to fit in there much better. So that particular dart needs to be augmented so that its binding to the target is substantially increased. But remember, it's not just binding that we care about. We are trying to make sure that upon binding, it functionally blocks the target protein from doing something that it was not supposed to do. Because all of this started with us identifying a target protein that needed to be suppressed in the context of certain disease. But in addition to binding and inhibiting the target's function, it also has to be soluble, it has to be stable, it should not be toxic. So now we enter the realm of multi-objective optimization. So here there are large compound libraries that can be synthesized around one particular compound, around your hit molecule. And so now the question becomes, which ones do I synthesize? So recall that compound synthesis is very expensive. So any computational method that can help you whittle down the list, that can help you rule out things that are going to be dead ends, is actually extremely valuable. So this is where many companies have contributed, both using computational chemistry methods and using deep learning methods. Schrodinger being a good example. Companies like In Silico Medicine have also incorporated AI at essentially every step. They've done target identification, protein structure prediction, protein binding site prediction, small molecule screening, and hit to lead optimization in an AI guided manner. There are so many startups and large pharma companies who have taken one step in this pipeline and built an entire department where computation and experiments talk to each other to achieve this hit to lead optimization, which is a very, very difficult problem. It's not a well posed problem because lots of data about compound properties are sitting inside different pharma companies, and efforts to make pool together the data have not yielded as much benefit as was initially hoped in projects like Synopsys in Europe. But these are the four stages of small molecule or protein engineering drug development that one should be aware of. And in each place, I went I described at a very high level, I would say, how computation and specifically deep learning based approaches have actually changed the way we do things. So today, no one attempts target identification without also having a layer to reason about images, or a layer that reasons about the language of proteins or about the language of DNA. So that is happening now. Similarly, no one does protein structure prediction anymore without the use of deep learning. And in terms of finding this small molecule, There are several different approaches to this. People see it. People see computation as aiding experiments, not really replacing experiments. So there are still companies that have large compound libraries on site that use robotics to do small molecule screening. That's how they get their hit molecule. But there are others that are employing an entirely computational approach to get that hit molecule. And so now a clear winner hasn't emerged. But what is clear is if you are stuck to only one strategy. So as a leader in your organization, if you adopt an exclusive experimental approach or an exclusively computational approach, you are perhaps sort of decreasing your chances of success. So now, as a case study and also to talk about protein engineering, I want to describe the modern drug discovery flywheel, especially as it applies to protein engineering. And I will use IQM as to explain how we think and how most of the field is now thinking. So the way we now think about this cycle or the flywheel, if you will, is continuously data has to be generated. So it all starts actually not with the model or the deep learning, but actually with ultra high throughput experiments. Because one lesson or one clear sort of insight that the field is collectively moving towards is that it is not the model that is going to give you a sustained advantage, but it is the data that is used to drive or build those models. So in a model, in a modern next gen synthetic biology startup, this is your typical setup. So you have ultra high throughput experiments that are conducted over protein libraries. So let me go back and explain what I mean by protein libraries. And also I use the word target protein before, and I'm using the word protein again here. So I mentioned that the target is a protein, it's a biomolecule, and we're trying to block it. Previously, we were trying to find a small molecule to plug into a little cavity on the target protein. But another way to block a target protein is to use another large protein to go and bind and block the interface that you're trying to block. Or it's also a way to just bind to the target protein and then use this binder to deliver the drug molecule. So in the context of antibody drug conjugates, for example. So it's a payload delivery mechanism, but ultimately it needs the protein that you're designing is the drug molecule that needs to go and bind to the target protein. So now previously we were making one protein at a time and testing, does it stick to the target protein? Does it stick to the target protein? But now the world has changed. Breakthroughs in a field called synthetic biology. So just like you have artificial intelligence, a parallel field is synthetic biology, where revolutionaries like Professor George Church and others in the last 2-3 decades have come up with technologies that allow miniaturization, multiplexing, and parallelization to happen on a scale that's previously unimaginable. So you may have heard that this fact is often stated that only in genomics or the cost of genomics, next generation sequencing was the Moore's law broken. So what that meant is technology was getting cheaper, roughly along with the rate at which computers are becoming more powerful. But suddenly the cost of next generation sequencing, which is the cost of sequencing DNA, actually dropped very precipitously. It became cheaper and much cheaper than expected. And that was possible because of breakthroughs in this field called synthetic biology. So in a modern tech bio startup, we have ultra high throughput protein library screening. In the case of IQM, for example, there are trillion protein libraries. So trillion different proteins can be made simultaneously. And the experiment that where we test one at a time is replaced by all trillion molecules trying to bind to a target. So this a similar concept exists in the small molecule world as well. That's called DNA encoded libraries or DELs, where there are as a large vial, billions or trillions of molecules, each having a small tag, and these small molecules are all competing for the binding site on the target. Similarly, here we have a large library of proteins, each with a tag. In our case, it's an RNA tag, and these molecules are competing for the target in the context of a vial. And this is where you can then subsequently use the tag, which is an RNA tag that each protein is attached to. By sequencing those tags, you can establish the identity of the protein designs that actually worked really well, meaning they bound to the target really well. So that's at the core. Ultra high throughput experiments are at the core at the start of this flywheel that powers the whole flywheel. So what does this lead to? So this leads to data that allows us to reason about the problem of interest. It could be protein small molecule interaction or protein protein interaction in the case of IQM. So we have these ultra high throughput experiments powering a massive in-house data set, a proprietary data set, which forms the core value. Now we come to model development. So now the field is evolving faster than they can keep pace with. As we speak, there are papers getting dumped on bioRxiv describing the next way to reason about protein sequences or about protein structures. And each company or each research group is trying to come up with the version of the model that works best for them. So either you use a LLM based foundation model that somebody like Facebook or Salesforce or Google have given us for free, or you build your own model based on your own data, which could be very different from what others are using, in which case you may just need to fine tune or build a completely new model from scratch. These models, so a model is nothing but something that allows you to reason about things that are not numbers. Protein and small molecules are not numbers. So a model helps you convert them intelligently into numbers in a representation that captures the underlying chemistry, physics, or biology. And now that you have numbers, you can then use these numbers in a second phase where these models power either generative design or they power a computational scorer that can go through designs and ascribe a score to each pair. So that's what happens in the computational protein design phase, where something like 10 power 11 designs are searched through by computationally. And now comes the kicker. So these models no longer just score, but they actually tell you what experiment to make next. So this is again a mindset change in the last few years, where deep learning, rather than being a scorer of ideas, has become the generator of ideas. So this is the computational protein design that happens. Once again, uh, Professor David Baker won the Nobel Prize for leading this field in this direction at the Institute of Protein Design. So we have computational protein design enabling the generation of a protein library, which then gets experimentally synthesized. And so we have a multiplicative effect between the number of ideas that were searched computationally that inform a more thorough exploration of a certain class of ideas, which is done experimentally. And so you can have this multiplicative effect of synthetic biology and artificial intelligence, enabling a much larger search space. You're searching through a space of molecules or proteins, or more generally, ideas that you're trying to, where you're looking for that initial molecule that did not exist. Remember, you need to do this because you're looking for a solution that did not exist before. You're trying to come up with something fundamentally new. So I know we are running out of time. So I'll use this opportunity to, I'll stop here and then leave some time for questions. But I'll use this opportunity to make an observation about how computation has served this field in the last 2 decades. So if you think about where it all started, computers were initially brought in because they allowed us to do things faster. So you could do more calculations. You could the number of things, the throughput of whatever you were trying to do, it was increased if you're doing it in a computational way. Then came the phase where you actually needed computers. There was no other way out, because now suddenly the human genome sequencing data came in, and we have 3 billion base pairs. You need a vast database to store it, and then you need to be able to reason about it. So it is no longer for efficiency, but it was a requirement, because the data that is being generated from sequencing, from ultra high throughput proteomics, from high resolution microscopy, these are all petabytes of data. So you need a number cruncher. Similarly, I mentioned molecular dynamic simulation. Ultimately, you need to do a lot of calculations. So you need a large amount of data over which massive amount of calculations are being performed. So you needed computation to enable that. Then came the phase where these deep learning models surfaced that could actually score the ideas. So you had a protein and a small molecule, and you could ask, is this what is the score? Like, what score should I give this molecule? Because I want the top scoring molecule. I can only afford to synthesize and make 10 top scoring molecules. So how does this particular molecule rank in the list of in my catalog or in some other way? So this is a where it captured a little bit of physics, it captured a little bit of chemistry, it captured a little bit of statistics, and it was essentially serving as a scorer of ideas generated by somebody else. But now we are in the phase where deep learning has become the idea generator. So we now learn, or what we choose to make and test experimentally in the lab is actually given to us or suggested to us by a deep learning um algorithm. So this is again, nothing works out of the box immediately. So it's not magic. It needs continuous fine tuning. We need to understand the mistakes it makes in its predictions, and then we need to correct for it. There are lots of breakthroughs happening. We are reasoning about protein at IKM, for example. We are reasoning about proteins using language models. We are also reasoning about proteins using structure-based models. There are so many breakthroughs that are happening where you can generate ideas. So there are breakthroughs reported where there are technologies such as diffusion based models where given an input we are able to come up with an output that complements the input. So essentially you can mask any aspect of the input and then ask the model to guess or impute what must fit in there. With that idea, you now have programs that can generate binders for any given target protein. You can generate binders for any given target small molecule. These are all idea generators, and this is the age that we are currently in. So, if your organization or your research group, or if what you're studying now seems to be that you're still focusing on scoring or data analysis or in just efficiency, it's time for you to start looking at what other organizations or other research groups are reporting about how deep learning can be used to generate what needs to be tested next in the lab. So I'll stop here with just one last thought, which is where are we headed next? Like, is computation done? So I won't open up the last image, but I will leave it to the audience to ask or to post in comments what they think is the next phase of deep learning. We already have a hint of what it is going to be, but I'll share that during the Q&A session after I see some of your responses on what you think AI is going to deliver next in this journey of drug discovery and development. Rohini, do we have any questions?
Yes, thank you so much, Venkatesh. I think it was such an enlightening and inspirational presentation. I think your expertise in combining AI with protein engineering has provided invaluable insights into the future of drug discovery, I must say. We now have some questions from, and it's time for a Q&A, as you rightly mentioned. We are going to, the first question is from Suchetna Gupta. She first of all says, lovely presentation, Venkatesh. And do you think with the advent of AI, ML, MD simulations will fizzle out? Or do you think MD and similar techniques will become more relevant because of the need of high quality training data? Do you want me to repeat it or it's good?
No, it's a very good question. I
Yeah.
can share about first of all the rationale behind the question. So molecular dynamics simulation, like I mentioned earlier, is based on the laws of physics. So you're trying to build in electrostatic potential, van der Waals interactions. So thereby you're getting the forces between atoms correctly, and that is what allows you to visualize molecules how they behave on a microsecond or millisecond time scale. But then the question becomes, is that necessary anymore now that we have AI? I would say it is very necessary because we will never know what is wrong if we don't have the ground truth. So the highest quality data is our experiments. But experiments are very limited in their ability to observe certain things because of X-ray, NMR, cryo-EM works for a very tiny fraction of cases. And there are some other techniques that you can use to collect data about proteins or biomolecules, but it's still very limited in explaining their dynamic behavior. So molecular dynamics simulations serve as one ground truth to try and mimic. So there are lots of ways in which AI has already entered the molecular dynamics simulation. The force fields, the forces used to represent molecules, previously they used to be regimented with certain equations from physics. Now we have come to the era of machine learned force fields. So like Suchetna said, if you have long trajectories of true MD simulations, you can then have machine learned force fields that essentially give you the same trajectory, but are able to perhaps do that computation much faster. There is also the associated field of instead of reasoning over numbers, you reasoning over functions. That's the Fourier neural operator work. Please look it up. So that's where we are trying to reason about the functions that operate on molecules or any physical phenomena, and that drives a larger scale. That makes a larger scale of simulations possible. So there are techniques already being researched about how AI and specifically deep learning for force field development and for reasoning about trajectories, where it can take us. So maybe the molecule is moving slowly when you, in a bucket of water as it is usually simulated, but maybe in some underlying manifold, it can be moved much faster. And so you move it along this the path that it is actually trying to move on, and then and wherever you need higher resolution, that's where you again blow it out into a full-fledged MD simulation and you run it there. So there are many techniques people are exploring, but it's a very good question. Whether
Yeah,
AI
amazing,
will
Venkesh.
replace physics? So my short answer is no, it will not.
Yeah, I think thanks so much for a detailed explanation on that. I hope Suchetna has got her answer. We have another question from Anmol and sorry, Amol. And his question is, how can one monitor AI's precision and sensitivity?
That's again a highly relevant question for biology among all fields, which is why there is a lot of, on one hand you have hype, on the other hand you have fear. In fact, there are people who are worried specifically about this combination of AI and synthetic biology, because what if you can very rapidly design toxic molecules or use it for some other nefarious purpose? So I would say, first off, we're approaching that time, but we are not there yet. But like in every other field, if we use LLMs, you get hallucination in text. You also get hallucination in molecules. But the good thing for computational biology is that we have a long history of computational chemistry, some ground truth tools where given a molecule, it can do some sanity checks on that. So we can compute solubility, stability, synthesizability. So we have filters that we can use to uh, to weed out molecules that are not likely to work. So even if it's a hallucinated piece of junk, there are ways of checking for other chemical signatures and discarding them. So this is one aspect of the question. The other aspect of the question was the error bars. So first off, if anything in biology, the error bars are larger because the data is more sparse. So yeah, unlike other fields where you can have a high resolution camera and collect infinite amount of data, or people for whatever reason have been typing in English online for millions of documents, so you can have a beautiful language model. We don't have that equivalent in biology. We have lots and lots of protein sequences. We have lots and lots of DNA sequences, but we don't have much about protein-protein interaction, protein small molecule data that's on any respectable scale compared to what a satellite imagery based company would have operated or have digested in the past. So we are still in the phase where we are very closely monitoring, what fraction of molecules synthesized actually work. So in fact, here more than error bar, what we compute is something called the hit rate or the enrichment factor. So let's say I synthesize 100, I synthesize 100 molecules at random and test them in an experiment. Maybe one or two will work for some random reason. But now let's say I take 100 molecules designed by my computational method, which may be AI, ML, deep learning, generated, searched. And then now I take those 100 molecules to the experiment. I don't expect all 100 to work. But let's say 10 worked. And previously, by chance, only 2 worked. So now I say, hey, I have 5-fold improvement in my chances. So I have enriched my hit list by a factor of 5, which is a very respectable amount. So we are very, very far away from being able to capture all of physics, chemistry, and biology on a computer. So we are happy if 5 out of 100 or 10 out of 100 work. So that's where the field is today.
Amazing. I think the next question is from Akshay Kumar Kamble. He has two. We'll take first question first. How language models are used for design? Yeah, that's the first part of the question. How language models are used for design?
Great question. So it is something that I wish I had made a slide on, but in the interest of time, I did not. So when we think about language models, and we say language models for biology, The first one that naturally comes to mind is maybe there is a LLM that has been trained on all the scientific papers. Maybe you ask what molecule should what protein should be targeted for some kind of cancer, and it gives you based on what it has read. So that is one kind of language model, but that just is a text summarizer, and that all of us are familiar with that application, and that is being used. That's being used to model patient clinical data, reason over doctor notes, electronic health records. But that is still not the breakthrough. So, in fact, one of the questions that I wanted to ask was everybody knows about protein structure prediction as a big breakthrough. But there is an equally important breakthrough that surprisingly has not yet been awarded a Nobel Prize or anything, but it is as big as protein structure development, and that is protein language modeling. So proteins, like I mentioned before, can be described as linear sentences in the chemical language. So every protein in our body, every three-dimensional complex molecule is actually a linear sentence in a chemical alphabet. That alphabet has only 20 different words, the 20 different amino acids. So what researchers decided to try about 5 6 years ago is see if, hey, let me take a GPT like model, and what happens if I train, if I just as input, instead of feeding it Wikipedia, if I just feed it my protein database, does anything meaningful come out? And so that is the big breakthrough. So what they discovered, multiple groups the same year discovered, was that protein language models trained just on protein chemical data, no English language, just protein chemical data, actually have given us a very powerful new representation of proteins. So now we can predict which two proteins are going to interact, which protein and which small molecule are going to interact. Is the protein going to be expressible in E. coli? Is the protein going to be stable at 35 degrees centigrade? All of these questions can now be answered because we now have a much more sensible representation of proteins over which we can then build, we can do supervised learning. So that's how protein language models have been primarily used. More recently, we have switched to the generative mode. So when you train a model, instead of just learning in an unsupervised manner um, what representation works for the protein, you can generate it in the same way that you do language to language generation. So you can have a certain prompt and teach it what the answer to that prompt can be. So in the case of proteins, let's say you're trying to design a drug for a small molecule, you can take as input a representation of a small molecule as text. So it turns out there is also a simple linear text-based representation of chemicals that is possible. So you have as input the prompt. You're asking this, drug GPT, hey, here is my drug molecule, make me a protein that binds to it. Or you can flip it and say, here is a protein molecule sequence that I give it as a language, and as output, give me the small molecule drug in a text format that will work on it. So this way, language-based generation, the same technique that is used for prompting, and for language translation, has actually been is being used successfully even at IKM for generating proteins that are likely to bind to the target protein. So language-to-language operations are very much hotly being researched and used.
Great. I think he has a follow-up question which says, also, can we predict in vivo characteristics of these molecules?
So that's the big unknown. So that's where I had that image of a cartoon of a mouse getting injected. So there, the issue really is that the lack of data. So there is only so much that we know about a large mammal, but what we can do is we can identify certain well posed sub problems over which we have successfully built models. So let's say the toxicity of a small molecule is because it actually binds to one of 10 different proteins in the body. Then what you can do is you can take that molecule and you can build a model that just predicts the binding of that molecule to each of those 10 different target proteins. Thereby, you effectively have a toxicity predictor, and then you can see how well it matches the in vivo data. Another in vivo property that is sometimes predicted is the ability of molecules to traverse membranes. Because when you take it by your mouth, from your stomach, it has to be absorbed into the bloodstream. So it has to be permeable across certain membranes. So certain experiments can be done. And if you have enough data for certain classes of molecules, you can predict this permeability. The last one is immunogenicity. So immunogenicity applies specifically to proteins. So when you give a protein as a drug, your body's immune system does not know that it's being given a drug. From its point of view, it will look at every protein that enters the body and ask, is it my own or is it an alien? Alien meaning not from my own body. Because that's how we defend ourselves from bacteria and viruses. We are able to detect that their genome is different from ours. So similarly, when an alien protein, which in case could be the drug molecule, enters, our own immune system may say, hey, this looks different from my own proteins. I'm going to grab it, break it, and dispose of it. So this immune system will flare up. That's called the immunogenicity problem. So clearly, when you're designing a protein drug, you need to be able to predict whether it's going to be immunogenic or not. And there again, a certain class of experiments have been performed. And so we have lots of data. And so to some extent, I would say to 10 to 20%, we are able to predict the immunogenicity. But really, we need data. So everywhere we go, the problem becomes, can biology now be scaled to the regime that will allow us to build a reliable model? So we may get there within the next 5 to 10 years, but not as of today.
Amazing. I think I think you're really explaining every question so much in detail. I'm sure, a person on the other side is going to be very happy. Just to let you know, Venkatesh, we have another three more questions, which I'm going to ask. So
Sure.
just letting you know about the time, right? So this is from Sono. And she is asking, can you discuss some successful examples of AI-powered drug discovery, such as identifying new targets or predicting drug efficacy? That's one part of this
So,
question. They have a follow-up, which I'll ask later.
sure. So I won't go into specific success stories, but I can mention a name of a few companies, and I invite you to do a deep diligence on those pointers. So I think I mentioned some of them before. Please look at in silico medicine, look at in vitro, look at recursion, and then there is a company in Israel called Biologics, which is, I think, brought the first AI designed protein drug into a human body. Then we have a few companies that are aspiring to get there, but they have not reached the clinic. So the lots of excitement around, DeepMind has started its own drug discovery company called Isomorphic Labs. Definitely something that we need to keep an eye on. And there are companies like Exientia in Europe, Benevolent AI. So lots of startups in this space, each looking for its big success story in terms of actually leading to clinical candidates. But I would say in silico medicine and recursion are farthest along in terms of having an AI-driven approach that has reached the clinic or is reaching the clinic.
Amazing. I think a follow-up question from her is, how are machine learning algorithms being used to analyze large data sets and identify potential drug candidates? What is the main key component to be in AI in drug discovery?
So here, really, to reason about anything, you need to first convert the input into numbers. So the big breakthrough that all of us need to become aware of is unsupervised representation learning. So if you have a long database of chemicals but with no other annotation, you would say, how can I learn anything from just chemicals? You need an annotation. It works, doesn't work, binds, doesn't bind. No, just the fact that it exists is the annotation. So you know that these chemicals are possible to be physically synthesized. That's why they exist. So there is a way which is very similar to how language is learned. So there is a simple fill in the blanks is really something as simple as fill in the blanks is how all of modern deep learning is happening. So let's say you have an image and you blacken out or hide a few pixels on that image. Our brain will have no problem in filling those pixels. We based on the color of nearby pixels, because we're always looking at blur images. We can, in our brain, we can create a sharper image. Similarly, this idea that if most of the data is there and some of it is missing, we are able to guess what needs to be there. That is the principle of masked language modeling. So to learn a language, to make a machine learn a language, you take a sentence, the apple is red, and then you show it only the apple is and ask it to guess red. But instead of giving it just one sentence, if you give it 100 million sentences drawn all from all over the web, the model will learn as it is trying, because the model during training is only trying to decrease the error. So it is adjusting so many different weights in the network so that the prediction error is less. So this is how all large language model training is happening, masked language modeling. So now the equivalent in biology, let's say you want to reason about proteins, the same concept is used. You take a protein sequence, but then you mask or you put a blank in certain spots, and then you tell the model your job is to help me fill in those blanks, and if you're correct, if you match the original protein sequence, you get a reward. If you're not, there's a penalty. The model learns to decrease its penalty by learning the underlying statistical distribution. So the statistical distribution is something that evades the human mind because it is far too complex. It is something that you have to reason about over millions of sequences. There are more than 100 million protein sequences are used in these training runs. And so there it effectively learns the residues that are important, the residues that may have co-evolved so that they always fold together. All of these are uncovered to the same principle. Then, when you're reasoning about small molecules, once again, it's the same thing. You suppress a portion of the chemical information and ask it to fill in the blanks. So this idea that you can learn from unlabeled data, because unlabeled data is plenty, labeled data is hard, meaning you need to know protein A binds to small molecule B with a certain strength C. That data is very sparse, but you have lots and lots of instances of proteins in nature, lots and lots of instances of small molecules as they have been physically synthesized. That gives you a rich representation and allows you, that is the basis of all of this utility in AI. Utility of AI in biology is that you can then do what is called few shot learning or one shot learning. Because if your underlying representation is so meaningful and rich, then with just a few labeled examples, you can make a much more sensible guess when you're given a new protein or a new small molecule and you're asked to predict the interaction. So at that point, you know so much about the underlying physics and chemistry indirectly that you can make a very, very sensible guess with just a handful of examples. You have 100 examples, you can make a sensible guess on the label for the 101st example because your underlying representation for the protein and the compound actually used 100 million data points. So I would say the short answer is representation learning is what is fueling the modern breakthrough applications of AI in biology.
Amazing. Thanks, Venkatesh. We have the next question from Venkatesh Kumar. He says, many drug candidates show promising in vitro results but fail in clinical trials due to unforeseen toxicity or poor pharmacokinetics. I'm so sorry. This is a jargon for me. I will try to pronounce it right. Uh, Pharmacokinetics. Got that right. How can AI improve in vitro to in vivo translation, enhance drug screening, and reduce late stage failures?
Again, it's a very
Yeah.
timely question, and it relates to that the central box in my five box journey, which is that where it is in a mouse, where we really, that's where we are lacking in data. So I answered this question in part before, but I think what can be done is it's the breakthroughs in synthetic biology that have to come to the rescue. We need to be able to get more data while sacrificing fewer animals. And only when we do that, or when we have a systems level understanding of the body where we can have a computational model that predicts what's going to happen, only then can we truly increase the rate of in vitro to in vivo success. Actually, that's something that's important for another reason. If you think about it from the commercial angle, so the way I presented it today, it felt as though the most important problem was target discovery, then came the drug discovery, and if you have those two, you've actually solved all of therapeutics. But unfortunately, not, because while you're predicting the first two or you're uncovering something new and you're improving its properties, we are still very far away from being able to predict the subsequent steps, and that's where the failures happen, and that's where companies lose a lot of money. So after having spent 10 years and $1 billion, something fails in phase 2. How do you recover that billion? You have learned a lot. Maybe some of this information, some of this knowledge is transferable, but very often companies get one shot on goal, and if that one project fails, they have to shut down the entire technology. Or unless you're a very, very big pharma company where you can afford to digest a handful of failures, because you can make up for it in the cost of in the price of the drugs that you sell. So it is in this light that it is important to understand that while I'm not an economist, while I cannot do a complete analysis of the drug pricing and related markets, it is important to understand that uh, drug discovery, if you want to find a new solution and take it all the way to the clinic, it is an extremely expensive proposal. And most of the things are going to fail towards the end of that journey. Early around those failures, you can always take a backup candidate. For example, even after 2 3 years, we have what are called development candidates that are taken to where you do very thorough preclinical study, but you always have backup candidates there. You can afford to have backup candidates. But once human clinical trials start, you've essentially bet everything that you've had. You poured everything that you had on one molecule, and that one molecule in its composition has to work all the way. So I think perhaps governments can interfere and enable. And as a society, we need to think about how to make clinical trials more viable, and how to do more animal studies in a less cruel manner. So one little development there is what are called organoid models. So here the idea is, well, I need to predict what's happening in a human, so I'm using I can't, so I use a mouse. But why? What if we could predict what happens in a mouse by some intermediate? So that intermediate is what is called an organoid, where instead of just growing some cells on a petri dish in a two-dimensional way, you grow three-dimensional mini organs. You can go grow a mini heart, a mini brain, and you can do it on an ultra large scale. So you start with the patient's stem cells or some other technology, and you can grow these miniaturized organs on which you can do tests that will definitely be much more predictive than your simple cell-based binding assay. So this field again is exploding thanks to breakthroughs in synthetic biology. So hopefully within the next 5 to 10 years, we will see a lot of animal testing replaced by organoid like technologies that are biomimetic technologies that are reasonable proxies for an animal model. And we are moving in that direction. But again, maybe another 5 to 10 years, and we'll be sacrificing far fewer animals.
That's amazing. I wish that comes, this technology is put to use as early as possible. The next question is from Bikram Ray, and he is asking, do regularity bodies like FDA has any role in computational biology?
So, in terms of computational biology, I'm not so sure. But in terms of computational methods that can be used for diagnostic purposes, there have been several very welcome breakthroughs, very welcome regulation. Where in the last few years there are computational methods that are now FDA certified for certain classes of diagnostics. Again, this has to do with the application of deep learning for reasoning about MRIs, X-rays. There are several startups in this space that have made significant impact, including ones like SigTuple in India, where they have uncovered several. Ultimately, it is in diagnostics. It's all about early diagnosis. The earlier you diagnose, the more favorable the eventual outcome will be for the patient. So more therapeutic options, longer expected survival, etc. But definitely there we are seeing a lot of AI based tools being approved. You may also see often you have these headlines where they do these blind tests where they take 1,000 scans, and they ask an expert radiology or radiologist to sort of annotate them. Then they feed that same input to this model that has been trained. And here and there, we see reports that a model actually outperforms a team of experts for certain applications. It's not universal yet, but that is something that's definitely going to happen in the near future. And the adage that applies to all fields also applies here. People who are radiologists who know to use AI are likely to replace radiologists who don't know how to use AI. So similarly, it applies to anything that we're talking about. So I think it is very important for all of us to become aware of the breakthroughs, see what is applicable for our organization or our research group, and then either bring in the talent or make a plan over a 2 to 5 year period to build that expertise in house, to recruit that talent, and also invest in the computational or other infrastructure. Because only uh, 5 years later to have this realization, it may be too late to catch up.
Absolutely. I think well said, Venkatesh. I think here we come to a end of Q&A session. Well, I think you did have a closing note, right? I think you wanted to discuss the last part about the presentation. There was a question, so you could take over on that.
Sure. So I had asked if those listening, if they had any thoughts on what they felt the next avatar of AI is going to be. I'd love to read some of those answers, and then I'll tell you what I think based on what I'm already saying.
Yeah, we have a response from Arun. He says it's personalized medicine.
Personalized medicine is a very good guess. Yeah, it is. It's so personalized medicine is where instead of making a drug for that works for the entire population, let's say I have a certain mutation, so I need a certain variant of a drug. And so, if we could rapidly sequence my genome, identify my mutations, and make a drug that works for me, and all of this has to be approved, there has to be a mechanism for doing this in a matter of months, then that's your personalized medicine. So, we already have a personalized medicine in the context of stem cell therapies. They are very, very expensive, but for a reason, because it's being done, all this R&D is being done for a handful of individuals. But really, the future that I'm seeing already is the one that is called the AI scientist, where AI is not just the idea generator, but AI actually does the literature search. It identifies hypotheses, but then it doesn't stop with telling you a hypothesis. It actually is able to handle. It's given control over certain laboratory equipment, so it actually goes to the next step of, let's say, synthesizing some proteins, or picking some compounds from some from internal chemical repository, and then it does those experiments, and then it analyzes the results of those experiments, and then it does this continues to iterate. So then it takes this new data, then it reads the corresponding papers, then it says, well, this may be the reason why it worked or it didn't work, and it comes up with a follow-up experiment. So the next no, the next decade is going to be about what we are seeing in other fields as agentic AI. Here it is the AI scientist, because we already have all the component pieces that I mentioned already exist. We already have companies like, for example, Future House, where they have digested all scientific literature. Now you can have a meaningful conversation about scientific topics with citations to papers. That's possible. Same with Perplexity. So then you have so you know that if you feed not just text data, but let's say you bring in genomic data, let's say you bring in phenotypic data from, let's say, microscopy images, let's say you bring in some proteomic scans, your spatial transcriptomics, you're reasoning about which proteins are expressed in which cell, and you have this information and the images, and you have the person's genome, and you have everything that is known about every protein all recorded in your model. Then you should be able to say this person has manifest this particular kind of cancer because it looks like KRAS has this particular mutation, because this image resembles that image in that paper. So now, but this person has this other mutation, which means the approved therapy won't work because that other mutation will hamper the ability of the approved drug. So I will start with that drug and make variations of that. So all of this reasoning that humans do, where we read papers, arrive at hypotheses, then we order the reagents, perform those tests, then we look at the results and that feedback. When we look at the answers, sometimes we don't know what question we are going to ask. But when we look at the answers, we are able to think of, hey, this is unusual in the following way, and this is unusual in this following way in one other paper or one other past experiment. So maybe these two are connected because I'm seeing an unexpectedly high value for this one marker or this gene that I previously was not looking at. Now suddenly it's suppressed. This combination is what results in that feedback to complete. So AI becomes the hypothesis generator. It generates the molecules to be synthesized. It performs the experiments and it ties the answers to go back and do the next set of experiments after doing the next research. So the AI scientist is the future that we are all eagerly awaiting. But don't worry, we are not going to lose our jobs. But we all need to know how to use the advances, the breakthroughs, so that we can do more and we can achieve greater success, because we are nowhere near curing or treating all diseases. So there are so many problems to be solved. So if a technology can give us some extra hands and help us get to a solution faster, we should embrace it so that as a society, we reach solutions sooner.
Absolutely, Venkatesh. I think well said. A lot of changes that's going to come up. I think we just got to embrace and upskill ourselves in the journey, right? So, yeah. So, Akshay, yes, this whole session is recorded, and you will be given a link to watch it again to everybody. Uh, Venkatesh, before we close, do you have any closing notes?
I want to thank you, Rohini and Priya, for giving me this opportunity to connect with the AI search community. I think your mission to sort of upskill people is, I think, is a very important one, because there is a group that is waiting for this syllabus to come to the school and college curriculum, but that's going to take 10 years, and by that time the field would have changed so much. So all of us have to go and find ways of educating ourselves. I'm no exception. So I come from the prestigious Indian Institute of Technology. I have my master's and PhD in computer science. But at one point in my career, I had to go to Coursera and a few other online sources to understand the breakthroughs that are happening, because the field is changing faster than we can keep track of. So it's very important for all of us to realize that no matter how good we are, we run the risk of becoming irrelevant if we are not aware of the breakthroughs around us. And it's very difficult, but on the other side, it's also possible to become a bit depressed, because the world is running too fast and everybody is reporting some breakthrough every day, and you can wonder, "Hey, how am I ever going to catch up?" So
Absolutely.
you do not try to master everything. So find one or two problems that are of a high importance or relevance to you personally, or to your current role, or to the role that you're aspiring to, and then just developing a solid base around one particular domain is what will allow you to will make you valuable, and will allow you to learn more things as they become relevant.
Thank you so much, Venkatesh. You know, unlike other certification bodies, AI certs actually offer AI certifications, which is more role-based, right? Not tailored to a person's profession. So AI plus healthcare is one such certification, and it's we change the curriculum on a daily basis, and we have subject matter expert at the back end to be in par with the technology and the changes that's happening. So we will definitely share all the details about AI plus healthcare to everybody. And whoever has attended today's session are also going to get 50% discount. So that's another. So we've created a coupon code in your name, Venkatesh. I think we're going to keep it as Venkatesh 50. So people are going to be using that code to upskill themselves. Well, I think from my side, thank you again for sharing your groundbreaking work and such deep insights on revolutionizing drug discovery with AI. I think your contributions are truly shaping the future of healthcare, Venkatesh. Thank you so much. And a big thank you to our audience for joining us today. And we hope the session has inspired you to explore the possibilities of AI in healthcare. Before we wrap up, a quick reminder, as I've already mentioned, the recordings of the webinar will be shared with you soon. Stay connected with AI Certs for more insightful webinars and resources by following us on LinkedIn or our website, which is www.aicerts.ai. Thank you once again, and have a great evening or a day, because we have a global audience wherever you have joined in from. Thank you so much for your time. And thank you, Venkatesh. I think it was a very insightful session today.